Hello, everyone. My name is Celine, and today I'll be presenting my master's research titled Genomic Insight into the Demographic History and Structure of the Grasshopper Sparrow. So the grasshopper sparrow is a broadly distributed grassland bird characterized by migrant and non-migrant populations. There are 12 recognized subspecies, but for my research, I focused on the 4 that breed in North America. Starting with the Arizona grasshopper sparrow, which is a partial migrant. Next is the highly migratory western grasshopper sparrow. Then another highly migratory subspecies, the eastern grasshopper sparrow. And finally, our very own Florida grasshopper sparrow, which is completely non-migratory. And so these 4 subspecies have experienced some alarming changes in their distribution over the last 100 years, but the most notable being observed in the Florida grasshopper sparrow, where they were once relatively abundant across 8 counties in central Florida. They are now restricted to just 4 and primarily found on public lands. So the combination of these different factors, differing geographic distributions, differing migratory strategies, as well as these changes in their population size, bring about questions regarding their diversity, their structure, as well as their demographic history. And so there has been previous genetic research in 2003 using mitochondrial data that looked at these questions, and they found a lack of differentiation. So they found a lack of differentiation between these 4 subspecies. And in their demographic history analyses, they estimated large effective population sizes as well as recent divergence times. And based on those results, they present this hypothesis that the lack of differentiation that they observed, or the lack of phylogenetic distinctiveness, was a result of shared ancestry as opposed to ongoing gene flow between these 4 subspecies. And so for my project, which that hypothesis sets the stage for, I address similar questions, but using a genomic approach. So looking at the entirety of the genome as opposed to just the mitochondrial genome. And my project can be split into 2 major parts, where first I assess genome-wide diversity and evaluate population structure. And then the second portion, which really digs into that central hypothesis, where first I infer changes in population size over time and test alternative demographic scenarios using coalescent modeling to see which model may best explain the demographic trend as well as the observed patterns of diversity and structure from the first section. So first, I assess genome-wide diversity using heterozygosity as a metric, and simply put, heterozygosity, the higher the value of heterozygosity, the higher genetic diversity is, and vice versa. And so here in the red box are the values I calculated for the different subspecies. And the first observation that we can see here is that the values are pretty uniform. But most importantly is that these values are low for what we would normally see for birds, and that they're consistent with estimates of heterozygosity for endangered species or species that have undergone major population declines. And so from those values, we can conclude that there is low genome-wide heterozygosity or low diversity across the different subspecies. And so next, I looked at population structure using FST as a metric. And broadly speaking, FST of 0 means no differentiation, and an FST of 1 means completely differentiated. And in this matrix, what we see here is that across the different pairwise comparisons, the different pairwise FST values, is that they are very close to 0, that they don't even reach 0.1, telling us that there is a lack of genetic population structure or a lack of differentiation between these different subspecies. And so now that we have an idea of diversity and structure, we can then move on to the demographic analyses, starting with inferring population size over time. And I did this using 2 different methods. First, to the left is a stairway plot. And stairway plot looks at changes in effective population size over time, but in more recent history, so within the last 10,000 years. And then again, I used a PSMC model. PSMC does the same thing as a stairway plot, but looking further back in time, so greater than 10,000 years ago. So together, these graphs give us this sort of complete picture of population size on the y-axis over time on the x-axis, from the last 1,000 or 100 years all the way back to before the last glacial maximum. And we can better visualize the population trend if we rearrange these plots like this, right? And if you follow this bird all the way to the right here, we see that grasshopper sparrows, the population peaked at around 90,000 individuals just before the last glacial maximum. And with the onset of glaciation and temperatures dropping, we see grasshopper sparrow populations declined down to just 20,000 breeding individuals. But then at around 10,000 years ago, with the onset of the Holocene, where temperatures are warming and grasslands are expanding in North America, there is a brief period of expansion. And then over the next several thousand years, at around 70,000 individuals, there's a period of stability. But then at that 2,000 year mark, there is an overall decline in grasshopper sparrow breeding individuals up until the last 100 years, all the way down to 8,000, to just 8,000. And so overall, we see that there has been this pretty significant decline in effective population size, with it really being more pronounced starting 2,000 years ago. So after inferring that trend, I then explored scenarios that may explain it, but this time just comparing the eastern grasshopper sparrow and the Florida ones. And in these coalescent models, I'm able to estimate gene flow, divergence time, and population size, which are all really important values to address that central hypothesis. And I tested 5 different demographic scenarios, 5 different divergence scenarios. The first one being no gene flow after split. Second is gene flow just in recent history after divergence. The third one is an early gene flow model where after split there's a level of gene flow and then reproductive isolation increases to where gene flow is stopped. And the fourth one is a consistent rate, ongoing rate of gene flow after divergence. And then the last one is a differential gene flow model where after split there is some level of differentiation or some level of gene flow, and that level changes in more recent history. And so the model that best explained the trend was the differential gene flow model. And in this model, divergence is estimated at 884 years ago, an ancestral population size of around 80,000 individuals, which is consistent with what we saw in the 2 previous plots. We can also get an idea of gene flow where there was an overall lower rate of historic gene flow after split, and then that rate increases in more recent time. And so these are what we're interested in when addressing that hypothesis from the earlier paper, where they found an ancestral effective population size of around 80,000 females. And they were only able to estimate this for females because they used mitochondrial data. They also estimated divergence of less than 26,000 years ago. And based on their hypothesis that gene flow, that there is little to no gene flow after divergence, based on their conclusions, which differs from what my data says, which shows that the divergence with no gene flow after split is the least supported model. And so we can come to some of these broader conclusions. First, my data does not support an evolutionary distinction between these different North American subspecies. Similar to that 2003 paper, it did not find a differentiation. However, alternative to their conclusions, my data shows that the lack of phylogenetic distinctiveness is more so a result of ongoing gene flow as opposed to the shared ancestry. And these are my collaborators. Thank you very much.